Oh, someone is in the waiting room. Okay. Hi, everyone. Welcome to Cell Cycle 2. Was it week 8? Topic 8?
No, week 9.
Oh, week 9. Okay.
I can't get along because
Oh, sorry. So today, because I think on Tuesday it was mostly content review. So today we decided to go over like practice questions to hopefully help you out with like either exams or also a lot of it is relevant to like the problem set. So if you have any questions, feel free to just stop us. Okay, so first one, true or false, it's okay for pre-RCs to reform during or after S phase. Yeah. Do you want to explain why?
Because you don't want the DNA to replicate anywhere.
Yeah, exactly. Because so the answer is false. And then if you do reform pre-RCs, then they can, what's it called? Like start replication again or fire replication again. And you don't want that because that could cause a lot of mutations or problems with like multiple DNAs and stuff like that. What about for 1B? There are 5+ ways for CDK to regulate pre-RC reassembly. I guess this kind of puts pressure on you because it's just immersive, but
I don't know. Are there like 3?
yeah.
Okay.
Nice. And if anyone on Zoom also wants to like participate with answers, feel free. But so the first one again was false. It's not okay because if your pre-RCs reform during or after S phase, the cell could potentially refire and re-replicate more than necessary. And then the 3 main ways for CDK to regulate pre-RC reassembly is phosphorylating the ORC, phosphorylating CDC6, and then phosphorylating MCM helicase complex. When you phosphorylate CDC6, it ubiquitinates it and then goes off to the proteasome and then it gets degraded. If you phosphorylate the ORC, the CDC6 is like inhibited from joining. So obviously that would prevent pre-RC formation. And then if you phosphorylate MCM, it gets exported out of the nucleus. So you can't have a pre-RC if none of the stuff required for pre-RC is around.
go sequentially because some of your bullet points. I don't know what you want.
Oh, okay. You're so good. Yeah, we can do that. Oh, okay. Question 2. What needs to occur at the beginning of M phase regarding spindle assembly formation? I feel like this question's a little vague, actually.
I was thinking probably like you have to depolarize the microtubule network. That we're going for.
Yeah, exactly. Hey, okay, maybe it's not that vague. Or maybe we're just on the same wavelength. Yeah, so I think this is kind of similar or like relevant to your problem set, but basically before M phase even starts, you already have a pre-existing network of microtubules, but in M phase, you have to have like spindle assembly. So first and foremost, you have to like cut down all of the microtubules that are already there and then use that tubulin that's now free to like create a new set of microtubules. And that can be done by like using a protein because when you cut a protein or cut a microtubule through the middle, I think we learned it in like week 5 or something like that. But that area is like GDP bound, and then that'll cause a catastrophe. Okay. Let me know if we're going too fast too. Okay, so this is just kind of like a label the area proper areas and like the difference between the different areas. So first we can start with this. This is pointing at like the 2 green cylinders. Do you know what they are? Do you know what they are?
Letters.
C.
They're all C.
Oh,
It's true.
It rhymes with, actually, no, that doesn't work either.
Centrioles.
Oh yeah, there you go. Nice. So these are the centrioles, and then the centrioles are within this orange circle. It also starts with sen.
Sen. Centro.
Yeah, this is also sen.
Centro.
Yeah.
Great.
You're so good. All right. Centrosome. So those are the spindle poles going to like the chromosomes. What about this? I think this is next. The red area. It's pointing to like these two ovals. Nice. And then lastly, the kinetochores are in what area? Awesome. Ace, yeah. Okay, so do you know the difference, like if you were to word the difference between kinetochores and centromeres?
Here's like the actual region and the part of the chromosome that attaches to sort of chromatids, but the kinetochore is like the protein complex that is off the kinetochore complex.
It's like you have the answer key in front of you. That's so good. Okay, this is a question. Yeah, so this is a question. I think this is like, I don't know if this was last semester. Was this your, okay.
Yeah.
I think this was a
I came to
question
your
that I
office.
found last semester. But these are like all the different types of generally important parts of the cell cycle. Cause like it can be split up like more or less. But I think overall there's 12. If you guys want to like take a minute or two and like think about it.
We could have used that marker.
Oh, we could have. Yeah.
People
Our marker was stolen.
I think people on Zoom wouldn't be able to see me.
That's true. I guess like if I
Oh, we
put
have
my
3
camera
people.
on.
Should tell Gianna the
Yeah.
answer. Say Gianna, and you should answer it from Nick Lang.
Okay.
Oh, were you guys logged in together for one of them?
That's mine. Yeah, Gianna's a different.
Oh.
But I know Gianna.
Gianna, you should answer this question according to Nick Lang.
I do
Oh,
know.
really, Nick? Okay. So,
We can take
Alright.
it one step at a time. What do you think is first?
okay. So first, I think, well, I'm trying to read all of these. These are a lot, but
Yeah.
G cyclins are made. I believe that's the first one, but I'm trying. Oh no, growth factor receptor binding to RTK
Yeah. Nice.
would be the first one. And then I would say G cyclins are made, but I don't know if I see another one before that.
Nope, you're good.
Okay. And then let's see. Oh man.
I know it's a little. I would have like a striker or something to like exit out.
Yeah.
Is that,
Oh,
can you
maybe.
do that?
Oh, I think I can annotate. Oh. Wait, is this a thing? Hang on.
I think it is.
How do I think I do? Oh wow. Okay.
Wow.
Okay, there we go.
Okay, we'll do. So this was, where is it? 1. Then this is 2.
Yeah, and then maybe CDK phosphorylation of ORC.
Before CDK.
Oh,
gets at-
okay, all right.
Something needs to happen, kind of like the point of no return before like we get into all the CDK stuff.
Ooh. Nick.
You want to help her out?
Sure. E2F.
Yeah.
activation.
Sweet. So E2F activation.
And then let's see. We need DNA replication initiation.
Yeah, replication initiation. Once we get through the next few, I can answer that. Yeah, so then replication is started. So if replication started, we don't want replication to happen again. So what would be something that we could do?
Oh, is it is it CDC 20? No. I think it's cyclin CDK phosphorylates we want, or is that that's the positive feedback loop though, right?
Yeah, the phosphorylating WEE1 is like has to do with activation. Do you remember like what's part of the pre-RC? Because that causes initiation.
Oh, is it CDK phosphorylation of ORC?
Yeah, because if it's one of like the 3 options that CDK can do. Because if it phosphorylates ORC, then it prevents like CDC6 from binding, and then you wouldn't have your pre-RC.
And then let's see. Would you need wait, CDC20 APC activation?
Ooh, that's APC has to do with like securin and those things. So that's closer to the end. Oh, this is a lot. Yeah, not yeah.
I wish I had the pathways in front of me. I could see it, but
Try like one more and then that'll be half of it and we can let Nick take the rest.
okay. I think the next one would be, but let's see. Okay, where's the last one we just did? CDK phosphorylation of
yes.
ORC.
Mm-hmm.
And then oh, it might be CDC25 dephosphorylates M-cyclin because then it can
Sweet.
go into M-cyclin.
Yeah.
Okay.
Nice. Thank you. All right. Your turn.
And it's like when CDK phosphorylates WEE1.
Mm-hmm.
In order for that to happen, CDK has to be active, right? So is there just like self-active CDC25 that can dephosphorylate and cyclin CDK to like get it started?
Oh, I see what you mean. I think CDC25 is just usually present in the
Oh.
cell. Yeah. Like nothing's you can inhibit it. Like that can be like something that Laura can throw at you, but in a typical cell, you just have like CDC25.
Okay.
Like you have phosphatases. Yeah.
The next one probably is the CDK phosphorylation of condensate.
Yeah.
Then probably
Well, let me know if I'm in your way.
is it CDC APC activation, and then securin degradation and shortening and settling.
Yeah.
That's good, guys.
Awesome. Yeah, that's awesome. Okay. I promise it's the trackpad. My handwriting is not this bad.
No, that was impressive.
Okay, how do I stop this? Oh. Okay. So that's just like all of it together. Because I think when I send out the slides, like the annotations have to be there. But yay, good job, guys. Okay. Oh, how do I erase that? Oh my gosh. Kind of chaotic today, guys. So sorry. Okay. Awesome. So a couple more questions for me and then Lily will take over. So what might happen to spindle assembly if egg5 is mutated or has a loss of function? If you guys like don't remember or like want a hint about what egg5 is or does.
Okay, I think I know this one.
Oh, go for it.
Okay. This is my comeback after the pathway.
You got it. You got it.
I know that egg5 helps pull apart the micro or like space them out. I guess I don't know how to explain it, but if egg 5 is mutated or has a loss of function, basically all the microtubules will collapse on top of one another. So the spindle assembly will and won't disassemble, but they'll just collapse.
Yeah,
Sorry.
so good. So just to give like a little bit more, if it'll come up, like details or context. Hello. Okay, so egg 5 is plus end directed, so that means it's like walking towards the center of the cell, so to say. And since it's plus end directed, that means it's outward pushing, so it's causing the spindle poles to be like further apart from each other. But like you said, Gianna, if that's not there, then there's only inward pulling, or like an inward force, and then that'll cause them to like become it's called like a monopolar spindle, but it's just the spindles end up like on top of each other. Awesome. Okay. True or false? When M phase is finishing, cells will be able to complete the cell cycle regardless of M cyclin CDK activity levels.
False.
Why?
It's like MCDK is just always doing stuff. You know, like MCDK. False because, well, MCDK is what determines if the cell is in end phase.
What does it
So,
do when it,
MCDK
yeah.
is targeting, it's target proteins and like phosphorylating them, so like the nuclear end below won't be able to
Awesome.
reform other things like that I can't remember.
That's okay. Nice. Yeah, that's pretty much it. So just to like expand on the other thing. So when you have M cyclin CDK in the beginning, like you said, it targets the nuclear envelope, specifically it's like the lamins that get phosphorylated. Nuclear envelope disintegrates or breaks down. And then the spindles also break down, and then also the chromosomes decondense. But at the end of the cycle, you want to have no more M cyclin CDK or like a lower deactivated level of it, so that your chromosomes can recondense, your nuclear envelope can reform, and that your spindles what's called reform as well, so that the cell is like finishing its cell cycle. Because if you constantly have a high level of M cyclin CDK, or like your M cyclin CDK levels don't go down, then you'll never be able to like finish your cell cycle, and it'll just keep going, and then that might cause like a mutated cell or something. Awesome. All you, girl.
Do you mind if I use your like forge?
Yes. Sometimes it's a little
It's okay, finicky computer.
yeah.
I'm not even gonna try to
It's
join from
Ace.
mine because it will just crash it.
You're so good.
immediately.
Please.
If this doesn't work, I can always just ask out loud.
It works, it's just so slow.
Like more Wi-Fi.
Oh wait, why does it say that there's oh, 2 seconds. I don't think it's refreshed.
Probably not.
I think it doesn't have the new slides that you have.
Oh,
Yeah. Quick intermission, guys. Perfect. One day my laptop's just gonna explode on me.
Oh, I know. Oh no, can you see that? If you saw that, no, you didn't. All right. So this is a true or false one. Kinases are enzymes that post-translationally modify their substrates by adding a phosphate group, which acts as an on switch for, the substrate.
That sounds right.
Sounds right?
Well, not always on, right? Sometimes
Okay.
it's like-
Yeah, right. So what would be an example of one that would act as like an off switch or deactivate a substrate?
When CDK phosphorylates V1.
Right, so WEE1 is a deactivating kinase. So normally when we see them, they are gonna be activating, but there are some examples where like phosphorylation will inhibit a substrate, so that is something to keep in mind, I guess. All right, so CBC 25, what would happen if there were a loss of function mutation? And if you want a reminder of what CBC 25 does.
If that happened, then it probably wouldn't be able to dephosphorylate the CDK cyclin.
Mhm.
So then there would be low levels of CDK cyclin activity in the cell cycle would progress.
Right, yeah, because it wouldn't be able to take off that inhibiting kinase, so it wouldn't progress into M phase. And then eventually, like if you get stuck for too long, the cell will die because the cell will try to correct mistakes that it makes when it's dividing, but eventually it's not worth the energy, or maybe the cell just assumes like whatever went wrong can't be fixed, so it will just initiate a programmed cell death pathway. And it would be similar if you had a gain of function in WEE1, so it's continuously phosphorylating mCDK. So you can have different mutations that will have the same effect in the cell. And here's a diagram, I guess, of that happening. So if we took away CBC 25, if we had that loss of function, you wouldn't be able to take off that inhibiting phosphate, and then you would stay inactive.
Can I ask something?
Oh, yeah.
Also like in the case that, say for instance, it was like loss of function, but then like the cell figured it out and like now you have CDC 25 again. But because it was like stuck in the growth phase or like the gap phase for longer, then your cell would be larger or smaller than usual. Would it be larger or would it be smaller?
Oh, wait, for the question you just asked.
Yeah.
CBC has a loss of function.
Yeah,
Then it would be larger.
exactly, because it just has a longer time to grow. Yeah. No, you're good.
Okay, so how would chromosome segregation be affected if GTP were replaced by a non-hydrolyzable analog? So one that couldn't be hydrolyzed to GDP.
I can answer that question,
Yeah,
I think.
okay.
think so.
That's all right.
You got it.
Give it a try.
So chromosome segregation would be affected because there probably wouldn't be chromosome segregation because you can't depolymerize the microtubules without having a hydrolyzable GTP analog. So without dynamic instability of microtubules, you wouldn't be able to segregate chromosomes or segregate them correctly.
Right, yeah, like when you have your spindles that are extending to try to find the sister chromatid that they're attaching to, like they don't know how to get exactly there, so they kind of just extend and retract until they find the site that they're supposed to bind on. But if you lose that ability to retract, then they're not going to be able to form their proper attachments efficiently or at all. So you need that dynamic instability for them to be able to find their attachment, and you would get stuck at least for longer than normal, if not permanently. I'm not like, you would have to see how it plays out experimentally, but you would get stuck for longer than you should in M phase, and it's possible that the cell would die if they don't because if your concentration of tubulin is too low to keep making microtubules because you don't have your existing ones depolymerizing. So yeah, that is exactly right. Oh no.
It's okay.
Okay, wait, I'm just gonna ask the next question. Okay. All right, so priming phosphorylation, how is it related to cell cycle regulation? Wait, actually, I can fix this right now.
Oh, I don't know if it'll
Oh, yeah,
correlate
no,
here
never
though.
mind. Yeah,
Yeah.
okay.
Do you guys know what priming phosphorylation means? Because I think sometimes in Clash he calls it something different.
Oh,
yeah.
sequential phosphorylation also. So just as a refresher, if any of you are not sure, that's basically when you need to have one kinase phosphorylate a substrate first before a second kinase in the pathway can act on it to phosphorylate it too. So how is that involved in cell cycle regulation?
Wait, what's the question again? Is it just how is priming involved in cell cycle
Yeah,
regulation?
like how does it regulate the cell cycle?
Oh, is this through like CDK being able to activate other proteins? It's like CDK. Oh, CDK can prime a protein to be activated or like deactivated.
Yeah, like pretty much. So
Okay.
CDK, because it's our like master cell cycle regulator, it's usually the one that comes first in a sequential phosphorylation. So you need to be phosphorylated by CDK before you can be phosphorylated by your next kinase. So the value that that provides is if you want to halt progression through the cell cycle, if you inhibit CDK activity, that will inhibit all of those like priming phosphorylation pathways instead of having to individually target all of the downstream kinases. You can just regulate it through CDK. Does that make sense?
I have a question about that. So
Oh, yeah.
would a protein or a kinase like CAC, like kinase activating kinase, would that be in the priming pathway? Like, how does that work? Is it everything but the proteins that activate CDK? Because like, how would CDK? I don't know. I get confused. Like, would WEE1 be in that pathway, the priming pathway?
Isn't that like
I think there's different, like priming phosphorylation is just like a term for something that can occur. So it doesn't always have to be CDK. So in your example of like CAC would be the priming one because you are phosphorylated by, oh, just kidding. What comes first, WEE1 or CAC?
oh my god.
I think WEE1 comes first because or do they come at the same time? I think they
I-
actually come at the
yeah.
same time.
Okay, that's what
Yeah.
I wanted to say.
So
but
maybe
so-
in this case, yeah, sorry guys. But I think in that case, I see where you're coming from, but since they happen at the same time, it might not be necessarily like
I don't think
priming.
it's considered priming because
Yeah.
like one isn't dependent on the other. One is an activating and the other is deactivating. So WEE1 kinase, that phosphorylation is just kind of holding it in the inactive state. And then once that phosphate is removed, it's like already ready to go, but one doesn't enable the other to happen. They just happen at the same
Okay.
time.
Yeah, and then you have to
Okay,
like
that makes
take it
sense.
away before it can actually happen.
Okay.
So that, I guess that's different than priming phosphorylation in the sense that you need one kinase for the other kinase to be able to act. And then your upstream kinase by like down regulating that because, you know, CDK is going to be your priming kinase often, then you can down regulate a whole bunch of pathways at once by targeting one protein instead of all of your downstream ones.
No, it's a little confusing.
Okay, so kinetochores, what do they do? And there are two things that I have, but you might be able to come up with others. How do they assist in chromosome segregation?
This is the one that I always think of, so I probably can't think of others.
They're what allow the kinetochore microtubules to attach to each sister chromatid to allow for proper segregation.
Right, yeah. So I mean, like the first thing would be that you have your attachments to your spindles, and then when you have your spindle attachment to your kinetochores, how does the cell know when all of them are attached?
I don't know.
But yeah, I mean,
That's
so
kind of
you're
right.
so true, actually.
Feel the tension.
Yeah.
Yes, exactly.
That's why we were like, I mean, that's not
Yeah.
wrong.
No, it's fine.
Exam
So-
2, vibes.
So MAD and BUB, the proteins, they will sense on the kinetochore that all of your sister chromatids have equal tension. And that means that from your two poles, they're being pulled on by spindles. So that is like your signal, like, okay, we're ready to go into anaphase. And then there is one more that I guess happens more when you're actively segregating.
Would it be like motor proteins combined to the kinetochores, like dynein?
Yep, that's perfect. So the motors will walk towards the minus end, so the far end, the polar end of your microtubules as they polymerize at the plus end, and that pulls your sister chromatids away from each other towards either side of the daughter cells that you have forming.
Yeah, almost like they're like eating the microtubules. Not that's not like what's actually happening, but like you can picture it that way if that helps. Yeah. Nice. Oh,
All right, one more. So, okay, if there were a gain of function in securin that made it unable to be degraded, where would the cell halt in the cell cycle?
It would hold like in end phase at the spindle assembly checkpoint because it can't let go of separate, so then separates can never cleave the cookies in so that the sister chromosomes will never separate.
It literally perfect, yeah. Metaphase checkpoint, so you would get stuck before you go to anaphase. And that's because when APC, CDC 2025, yeah, CDC 25 APC complex ubiquitilates securin, it has a gain of function, so it won't be degraded. It will stay bound to separase. It will kind of just like sequester it, and then cohesin would not be cleaved, so your chromatids can't separate and anaphase cannot happen. So the cell would probably also die. A lot of these, just like your final conclusion is, all right, well, you mess it up, the cell's gonna die.
I was right that my problem's dead. What would happen if it dies?
Yeah. Yeah, no, I feel like a lot of these you can always wrap up with the cell would die or
Yeah.
would lead to cancer.
yeah.
Yeah, so yeah, that's great. Great.
So that's all we had for questions. If anyone had questions about specifically like anything that we covered from the questions that we had right now, I guess that was a lot of questions. Does anyone have questions on what we could just covered? Okay, if not, I'm gonna stop the recording, but we can go over questions that people might have generally in class. How do I stop recording?
Uh,
Oh my god.
wait, why do I
At
not
the top
know?
left, if you press.
Oh, wow.